Hey guys, it's Mr. Boyle. Trying to do a few videos for you guys on some of the tougher concepts, more important. This one's on protein synthesis, and kind of a couple steps there. I'm going to talk about the RNA molecule as well, and see if we can get this to work. If you need some help, text me, email me, etc. And I'm going to do one on what's called the codon worksheet, which will kind of be the next video, and we're going to kind of touch on it just before we finish here, this one. So protein synthesis occurs kind of in two steps. One's called transcription, one is translation. Protein's main job, most important job, cell's most important job is to make proteins. And those proteins come from genes. Remember, a gene is enough information for a cell to make one protein. For example, the pancreas, one of the proteins that the cells in the pancreas make is insulin to help regulate your blood sugar. Now, in the transcription part, it occurs in the nucleus, and the DNA section opens up where the gene is, let's say for insulin, and an mRNA, a messenger RNA molecule is made. I'll talk a little bit about RNA in a sec here. So the messenger RNA molecule is made by matching up to that DNA strand, and that gives it the information to make the insulin. So a messenger RNA strand is made from the gene segment of DNA. So I just made a short segment of DNA here, 12 letters, ATG, GCG, TAT, GCA. And then with the RNA, you're going to see that there are U's called uracil instead of T's. The other letters are the same. The A, the G, and the C are the same, adenine, guanine, cytosine, but there's no thymine in RNA. There's one called uracil. So A matches to U, T with A, G with C, G with C, C with G, and so on. So when we look at RNA, it is single-stranded. All right, DNA is double-stranded, it can open up. RNA is just one strand. It has uracil instead of thymine, so A with U, all right, and the match. And the sugar in DNA, it's deoxyribose sugar. In RNA, it is just a ribose sugar. It's a little bit simpler, all right? So making that mRNA molecule in the DNA, or attaching to the DNA, that's in the nucleus. Then that mRNA strand sneaks out of the nucleus. There's little holes called pores in the nucleus that the mRNA strand is small enough to sneak out of there. It then heads out to the ribosome. And at the ribosome, it does a process called translation. So the mRNA strand travels out to the ribosome. It literally attaches to the outside of the ribosome. It has the code now that is needed to make that protein. The tRNA now, that's called transfer RNA. It's out there in the nucleus. It has little amino acids attached to it, and it attaches to the correct amino acid. That's the important part. The tRNA molecules brings the amino acid, which are the building blocks of proteins. If you're working out, you know you need amino acids or protein after you work out to build those muscles back up. Right? Attaches to the mRNA. It attaches there, so there's a connection, and that's based on the letters. Right? The A's again match with U, G with C. The amino acids will link together. They're like little box cars on a train. It's one way to look at it, or little paper clips hooked together. And if you get enough amino acids, you then have a protein. Now, some proteins are like 7 amino acids long. Like insulin's a pretty simple one. Some are literally thousands of amino acids long, and they have to twist and turn into a specific shape. So some proteins are easy to make, some are not. Now, if we look at the amino acids, they link together, they're held together by what are called peptide bonds. All right, and once that protein is made, the cell can use it, or it sometimes goes into the blood to be used somewhere else in the body. Like, for example, testosterone is made in the testes, but it's used in other places in the body, and for example, in helping to build muscle. Testosterone's a muscle builder, helps with muscle building. Okay? Now, so we look at the lettering here. DNA has the code, right? The same code as I had on the other board. RNA has a code that matches to it. Then tRNA matches. So when we have a DNA of GCG in 2 steps, that code is back to GCG. And that tRNA molecule brings with it an amino acid. In this case, it's one called arginine. Right? There are 64 codes possible in DNA. There are 20 amino acids. So some codes or some amino acids have more than one code that can bring that amino acid. That's why some mutations are no big deal. As long as the code brings the right amino acid there, there's no issue. Okay, so I will go over the amino acid chart, all right, and how to do that in the next video. Good luck. Give me a call, text me, email me, FaceTime me if you need any help. Good luck.